And then for the last talk in our session this afternoon, I'm very pleased to welcome Dr. Clark Russell from the University of Edinburgh. Clark's a clinical lecturer and his research is focused on macrophage-mediated responses to infection. But over the last 18 months, he has done extensive work on COVID-19, and I believe he'll be talking to us about treatable traits in COVID-19. Clark, welcome. Thank you.
Thank you very much indeed. It's a great privilege to be here to talk about some of the work from ISRIC 4C, and in particular to do so on behalf of my friend and boss, Professor Kenneth Bailey from the University of Edinburgh. So ISRIC 4C was set up in February 2020, and the leads of the programme are Kenneth Bailey in Edinburgh, Callum Semple in Liverpool and Peter Openshaw in London. This is a collaborative consortium recruiting patients at about 260 UK hospitals. To date, we have clinical data on over 250,000 hospitalised people with COVID and biological samples in over 2,000, which are collected in outbreak labs in Glasgow and Liverpool, and then sent to downstream research groups for downstream analysis. In addition, I'll be talking about some work from the genomic study, which is a genetic study of people admitted to intensive care units, also led by Kenneth Bailey, and a post-mortem immunology study led by us in Edinburgh, recruiting people who had died due to COVID-19. It's known as the ICECAP study, which is a dreadful acronym, which you can ask about later. So this is the broad approach that we've spoken about for some time for trying to integrate diverse data sources in order to better understand infectious diseases at a systems level. And this pinpoints where the different data sources are coming from that I'm going to talk about today. So we have clinical data from the ISRIC 4C study, over 250,000 hospitalised people. We have host genetics from the genomic study, and I'll be talking about results of microarray sequencing from 2,200 people with COVID-19 admitted to UK intensive care units. We have data on the host response, and I'll talk about inflammatory mediator measurements in plasma from the ISRIC 4C study in hospitalised people with COVID. And then finally, details at the tissue level of host pathogen interactions of host and COVID in post-mortem tissue analyses from the ICECAP study in Edinburgh. And the goal in trying to integrate these data sources and cross-validate host targets is to identify the right patient and the right target. And that's the concept that we're interested in terms of stratified medicine and treatable traits. So to talk about the terms that I'll use, I'm not saying that these are the correct terms, but the terms that I'll use in my talk. When I talk about a subgroup, who in a group of people who are related by the fact that they have shared clinical features. An endotype means, to me, a subgroup of people who have a shared biological process. So that's one mechanistic step further. And then finally, treatable traits, we're talking about the components of inter-individual variation that are differentially responsive to different therapeutics, and will be the focus of what I'm trying to talk about. So there's this very thoughtful viewpoint in JAMA, and it's got the very provocative title, Phenotype Anarchy, referring to COVID-19. And this refers to the fact that with the large number of multi-omic studies that have characterised host pathogen interactions in COVID, there's a lot of data, and they say the degree to which this diversification is relevant to bedside treatment is largely unknown, and that's a very fair statement to make. So there's reason to think that actually finding treatable traits, so finding components of variation that are therapeutically relevant, might be quite difficult to do in COVID-19. So if we think about the relative heterogeneity of different infectious diseases, sepsis is probably a broadly unrealistic term to define anything precisely. Host variation is a constant. In sepsis, the organ systems involved are diverse, any organ system, and the range of pathogens, of course, is also huge. So the term is totally agnostic to the pathogen. Then, thinking about a disease which, as an infectious diseases person, interests me a lot, Staph aureus bacteremia, this is probably slightly more homogeneous, but still pretty heterogeneous. So these metastatic complications of Staph aureus affecting just about every organ system in the body are all, unfortunately, common and never a surprise when they arise in the care of somebody with the Staph aureus bacteremia. So organ system, very heterogeneous. The pathogen itself, although it's the same pathogen, phenotypically different Staph aureus isolates can vary in quite important ways. So methicillin susceptibility, which dictates antimicrobial choice, but also the presence or absence of clinically relevant toxins, so the Panton-Valentine leukocidin toxin or toxic shock syndrome toxin. Becoming slightly more homogeneous is community-acquired pneumonia. So at least we've now narrowed it down to a single organ system, broadly, the lung. But then the pathogen is very diverse. We only actually know the pathogen through a microbiologic diagnosis in about a third of hospitalised people with community-acquired pneumonia. It can be conventional gram-positive or negative bacteria, it could be atypical bacteria, it could be viruses, in rare cases could be fungi. And then we have COVID, which I would contend is probably the most homogeneous out of all of these. Again, it's broadly the same organ system that's affected. It's the lung, and that chest X-ray is probably depressingly familiar to many people in this room. But also the pathogen, we know what the pathogen is, it's the definition of the disease, and also broadly, when in comparison to other pathogens such as Staph aureus, there's probably a bit less phenotypic variation. So that, of course, is providing the devil's advocate's perspective on it, and we know that there is significant heterogeneity within COVID-19. So this is the first, one of the first analyses from routinely collected clinical data from the ISRIC-4C study. This is unsupervised clustering of symptoms on presentation to hospital. You can see there's a core symptom group, which is most common, of respiratory symptoms, but also, for example, in about 5% of people, there's an enteric cluster of symptoms. So these people have diarrhea and vomiting in addition to their respiratory symptoms. And the unadjusted 30-day in-hospital mortality for this group is substantially lower, shown in the Kaplan-Meier plot. Another differences with this group is that people in it are much younger than the rest of the cohort, than people presenting with typical symptoms. Looking more broadly at a cohort of about 20,000 people hospitalised with COVID, this factor of age comes out as being a very strong predictor of in-hospital mortality. So there's a stepwise substantial increase with increasing age, and it has the largest effect size out of all the other comorbidities. Trying to refine that, a prediction model was developed to predict in-hospital mortality, the 4C mortality score. And this starts to allow us to stratify patients into subgroups based on their risk of death in hospital. And again, age, as you increment through the increasing age brackets, you get the most points as you go up, but also this time we see starting to get some insight into biology. So for example, CRP. Increasing CRP gets you more points, and the score is associated with increased in-hospital mortality. Now, we now know that inflammation is causal in death in COVID. So there are now several randomised trials and meta-analyses that support the use of anti-inflammatory glucocorticoids, which improve survival in hospitalised people with COVID. And we'd contend that that's evidence that inflammation is causal in death due to this disease. And that, in itself, thanks to the pragmatic design of some of the trials and the subgroup analyses, already allows some application of subgroups in clinical practice when looking after people with COVID. So from the recovery trial, we know that people who required any degree of respiratory support derived benefit from dexamethasone, and that was greatest in people who were receiving invasive mechanical ventilation. But we also know that people who did not require any respiratory support derived no benefit and potentially suffered harm due to dexamethasone. So at the level of the very gross level of severity of lung injury, there are already some subgroups that are clinically relevant. So when trying to refine this to understand what might actually be some treatable traits in COVID, we asked the question, what is driving this inflammation which is leading to death? Is this virus-driven? Is it a direct effect of SARS-CoV-2? Or is this host-driven virus-independent immunopathology? And this was the core question behind the IceCap post-mortem study, which we conducted in Edinburgh. Sites in the body were systematically sampled, and viral detection was achieved through Arctic Protocol PCR. This was done by Julian Hiscox's lab in Liverpool. Arctic Protocol PCR confirmed by sequencing, and also detection of viral subgenomic RNA as a proxy marker for replication, and then by immunohistochemistry detection of viral glycoprotein. And we were able to show that there is widespread distribution of the virus at the time of death, so up to 42 days after symptom onset. But at the same time, there is a disconnect between the presence of inflammation. So samples from the same blocks of tissue were used for histologic analysis and scoring, semi-quantitative scoring of presence or absence of inflammation by organ-specific expert pathologists. And there is a mismatch between the presence of virus and the presence of inflammation, with inflammatory changes largely restricted to the lungs and the reticular endothelial system. So in the case of the samples that we took, the spleen and the bone marrow. And this heterogeneity exists at the within patient level as well. So each cell in this sunburst diagram represents an individual tissue block, and they're divided into the individual patients. And you can see that on the outer ring, which is presence of viral RNA, and then the ring inside of that, which is the presence of diffuse alveolar damage, you can have viral presence without any inflammatory changes, and you can also have inflammatory changes without any viral presence. So briefly, we interpreted that as evidence that in fatal COVID, occurring clearly late in the disease process, that virus-independent immunopathology is a pathophysiologic process that's happening. So now to address some of what might be the pro-inflammatory mechanisms underlying that. When studying the immune cell infiltrates in the lung in fatal COVID, we did multiplex immunofluorescence to characterize the immune cell populations. Something that was interesting we saw was a mononuclear cell pulmonary artery vasculitis, but more broadly what we saw was a parenchymal expansion of macrophages and monocytes, so CD68 positive cells with variable MRP8 positivity that were all mononuclear cells. So macrophages are monocytes that were substantially more expanded than any other immune cell population. And on the multiplex immunofluorescence image, the CD68 marker is in green. And others have shown that this expansion of macrophages in the lung parenchyma is specific to COVID and distinct from fatal influenza, bacterial pneumonia, non-selected ARDS, and health, which supports a specific role, we think, for macrophages in COVID-19 pathogenesis. We used these same tissues from the same well-characterized patients to undertake tissue proteomic analyses to try and understand some of the humoral mediators of inflammation at the level of inflamed tissues. And this was done in collaboration with colleagues in Olink. I'm just going to pick out two proteins of interest from the differential abundance analyses. In the lung, MCP3, so macrophage monocyte chemoattractant protein 3, and in the spleen, MCSF, or CSF1. We then compared our tissue proteomic findings to a publicly available data set of plasma proteomics and found some overlap, including MCSF and MCP3. We then used unsupervised clustering with two different techniques to try and understand if there were any distinct clusters or endotypes within our proteomic data in the lung, and we identified that there were. So first of all, the controls separated quite gratifyingly. And we identified two distinct clusters of the COVID-19 pulmonary proteomes based on the proteomic data. And when we then looked at some of the associated metadata, some interesting differences emerged. One cluster had a significantly longer illness duration at the time of death. They also had significantly less detectable virus at the time of death, and they had more severe histologic inflammation at the time of death. We did differential expression analysis in these clusters and interestingly found that, so we named these clusters the late inflammatory cluster, so that's the late cluster without virus, and then the early viral cluster, which was the cluster with detectable virus and the shorter illness duration. And in differential expression analysis, interestingly, we found that interleukin-6 was at a significantly lower abundance in the late inflammatory cluster, or conversely, a significantly higher abundance in the early viral cluster. Shifting to a different data set now, this is plasma inflammatory mediator data from the ISORIC-4C study, measured by Ryan Thwaites at Imperial College. And broadly, the point of this slide is to show that most pro-inflammatory mediators scale with disease severity. So I don't know where the axis labels have gone. They were here this morning, but on the x-axis, it works up from, so green is uninfected healthy controls, pink is community cases, and then works through the WHO ordinal severity scale. No oxygen, oxygen, non-invasive ventilation, invasive mechanical ventilation, and fatal disease being in red. And so disappointingly, the labels aren't there either. So in the inflammatory column, two of the mediators are interleukin-6 and GM-CSF. We then did some clustering analysis on these inflammatory mediators and identified 4 distinct endotypes. So calling them endotypes because we think that we understand a little bit about how the mechanism differs between them. One of which being what we call the hyperinflammatory endotype, which was associated with increased in-hospital mortality. And 2 of the mediators that were contributing to this clustering were GM-CSF and interleukin-6. Principal component analysis to look at the contributors of the variance when assessing for differences in disease severity. So again, looking at the WHO ordinal scale, again identified interleukin-6 and GM-CSF as significant contributors of the variance. And finally, network analysis identified interleukin-6 and GM-CSF as highly connected hub proteins, which the presence was correlated strongly with large numbers of the other pro-inflammatory mediators that were measured. And finally, there are differences and similarities with influenza A. So we were able to have access to archived plasma samples from people with fatal influenza A from the MOSAIC study. And Ryan was able to measure some of these mediators in those samples and show that interleukin-6 levels were actually the same, which was interesting when thinking about therapeutics, but that the elevation of GM-CSF in COVID seemed to be specific and wasn't observed in influenza A. We had access to plasma viral loads, quantitative viral loads in plasma, but shown here as a binary presence or absence. And these were measured close to hospital admission, so saying that these are quite early samples, about 7 to 10 days after symptom onset. And the presence of virus or viral RNA in plasma is associated with significantly higher interleukin-6 concentrations early in disease and also substantially increased in hospital mortality. We plotted some of these inflammatory mediators against the illness duration at the time that they were measured. And for some of what might be considered the more proximal upstream inflammatory mediators, we didn't really see any changes to GM-CSF and interleukin-6. Whereas for and in fact, they probably started to decline a little bit, whereas more downstream markers of tissue injury, such as D-dimer and NRAGE, didn't decline and trend that they increased. So combining the interleukin-6 findings, so the lack of an increase as disease time increases after hospitalization, the finding that it's associated with plasma or anemia early in disease, and the findings from the tissue proteomics that it's interleukin-6 is higher in a cluster of people who died early with detectable virus, we think that this might provide a sort of a comment about therapeutic relevance or treatable trait, and explain one of the findings from the clinical trial of tocilizumab in the recovery trial, where obviously a beneficial effect was seen overall, but when tocilizumab is started in people already receiving invasive mechanical ventilation, so at a later disease stage, this benefit is not seen. And it may be that interleukin-6 exerts its pathologic role earlier in the disease process, and therefore once somebody is admitted to hospital, they're probably already at the stage where interleukin-6 activity is maximal and most relevant, and therapeutic inhibition might make more sense earlier, explaining why the lack of benefit is seen when they're already ventilated. Thinking about the histologic finding of monocyte macrophage-driven lung inflammation, this result is from a transcriptome-wide association study as part of the genomic study. So here we took variants that were known, so genetic variants known to have an effect on transcript abundance, and from that inferred the direction of effect to the genetic variants that were associated with the phenotype, in this case the phenotype being admission to an intensive care unit due to COVID-19 as opposed to matched population controls. And one of the, and this is from the GTEx lung tissue database, one of the genes whose expression is increased or inferred to be increased in COVID-19 cases is CCR2. CCR2 is a receptor for 2 macrophage monocyte recruitment molecules, CCL2 and MCP3. CCL2, we've shown in the Isseric plasma mediator data, is increased with disease severity, and MCP3, we've shown in lung, and the overlap with available plasma data is also increased in COVID. So we propose that this, accepting there will of course be multiple axes and mechanisms, perhaps one axis involves activation of the monocytes by MCSF and GM-CSF in the periphery, and then recruitment to the lung centred around CCR2 activation by MCP3 and CCL2, with potential for genotype-dependent effects on CCR2 expression contributing to severity. A treatable trait here might be GM-CSF. So the Kaplan-Meier plots are from the GSK OSCAR trial of a monoclonal antibody against GM-CSF. Overall, no benefit was seen, but in a pre-specified subgroup analysis of people over the age of 70, benefit was seen. And I believe that they are continuing to recruit into that age bracket. And in our ISRIC4C data, when GM-CSF is stratified by age, there's quite a stark difference with much higher GM-CSF and detectable GM-CSF seen in people in the age bracket that derived benefit in that subgroup in the clinical trial. So finally, a comment about host defence and antiviral mechanisms. This is returning to the genomic GWAS, so 2,200 critically ill people with COVID-19, and this is the results of the GWAS. These are variants in genes that are associated with phenotype, and 3 of them are in interferon signalling. So there's IFNAR2, which is the receptor, there's OAS1, and there's TIK2, which are downstream signalling components. We know from multiple in vitro studies that interferon induction appears to be defective in SARS-CoV-2, and from this work, and again, I don't know why the axes have vanished, that in contrast to many other inflammatory mediators, interferon alpha in blood does not scale with disease severity, nor is it increased in people who have plasma anemia at time of hospital admission early in disease. And again, from others' works, this is from Jean-Laurent Casanova's group, that there are neutralizing autoantibodies present against type 1 interferon, so against the ligand, in people with life-threatening COVID. So in that study, about 100 out of 1,000 people. So we propose a sort of very simple mechanism for how these things might fit together. That pre-hospital, probably the primary defect is impaired viral control. And there is genetic evidence for defects in signaling, and then evidence from autoantibodies for loss of the ligand or for loss of function of the ligand, leading to widespread viral replication. Inflammatory priming probably occurs early, for example, the association of interleukin-6 concentrations with viral presence, and the response is probably quite primed by the time somebody gets to hospital, in that there isn't a further increase in these mediators as duration after hospitalization increases. And probably during this time, the relevance of the virus itself in the process is diminishing, consistent with the finding that at late stage in fatal disease, tissue immunopathology appears to be at least partly virus independent. And then there's the macrophage axis, which I've discussed. And that might give some insight into the findings from 2 trial results of exogenous interferon. So first of all, there's the WHO Solidarity trial of hospitalized people. And this plot shows the in-hospital mortality of interferon versus usual care control. There's no statistical difference, but clearly interferon, there's a signal that exogenous interferon is associated with increased mortality. And then published more recently is the ACTT3 trial, comparing this time exogenous interferon with remdesivir against remdesivir and placebo. And in the less severe subgroups, they found no difference, but in this subgroup of people who were receiving high-flow nasal oxygen at the time of recruitment, they see clear evidence of harm in the people who received interferon. They see an increased time to recovery. So it may be that simply that providing exogenous interferon in hospitalized people is too late. By that stage, it's a virus-independent process, and the pro-inflammatory effects of interferon are unhelpful. It may be that, of course, the people who are defective in interferon responses are genetically unable to respond to the ligand because of genetic defects in the signaling pathway, but that's much more speculative. So finally, trying to return to the thesis of the talk, treatable traits, considering the host, first of all, so host-driven inflammatory processes that are damaging in COVID, we know that monocyte macrophage recruitment and activation is abnormal and probably specific or specific-ish to COVID. GM-CSF seems to have a differential effect with age that might be therapeutically relevant, and CCR2 expression is definitely associated with genetics. Thinking about interleukin-6 signaling, this time the treatable trait may not be a fixed one, such as genetic or age, but the point at the person's disease trajectory that they are at, an earlier inhibition in hospital might be helpful. In terms of defective host defence and defective interferon responses, it's probably a bit more difficult. We know there's genetic evidence of defects in signalling. We know that there's evidence from autoantibodies of loss of functionality of the ligand. Is the lack of benefit, or in fact, is the harm from exogenous interferon related to the time at which it's given in hospital? Would it help if it was given pre-hospital? Or is the issue the supraphysiological doses that are presumably being given? But either way, there's probably a requirement for alternative antiviral strategies to interferon, of course, and alternatives to remdesivir. So with that, I would acknowledge the massive number of people that have been part of the consortia that have generated the data that I have spoken about, in particular to Kenneth Bailey, Peter Openshaw and Callum Semple, and the funders of this work, and also thinking specifically about the post-mortem study, the generosity of the next of kin who provided consent for the research post-mortems was often quite humbling. Thank you.